Hi, my name is Nabila Bouatia-Naji. I'm a research professor at the INSERM, Paris Cardiovascular Research Center and Université de Paris. I'm going to talk to you today about how genes influence the health of arteries, results from our study about DNA of FMD patients. So just a reminder, DNA is the code of life. This is an information that you're probably aware of. DNA is stoked into cells. Cells are having information of DNA is part of the nucleus, and the nucleus contains chromosomes, which are a combination of proteins and DNA. And DNA is the molecule where the genetic information is stored. It is organized into genes or segments of DNA. So information of the segment of DNA is about the succession of nucleotides that are called C, G, A, and T. And this information is then copied into a RNA, and this RNA can produce a protein. And those proteins are the elements that build a cell, and the cells build the organs, and then the organs make the body of the human beings, but also all kinds of living organisms like plants, bacteria, etc. In our study, we were interested in understanding how genetics can influence FMD. For that, we used almost 1,500 DNAs from FMD patients. So maybe one of your DNA is part of this study. So thank you for contributing to our efforts. DNA from those patients were collected from blood. From blood, we extracted the DNAs, and it contained exactly the same information that may be part of the artery and the heart and everybody, a part of the body. Then we did exactly the same thing from more than 7,000 DNAs from a group of controls. These are individuals where we know that fibromuscular dysplasia is probably not present. So we compared the DNAs from patients, the cases, and the controls. And we analyzed what is the frequency between the cases and control. And we were interested in variation with higher frequency in cases compared to control. We did this for 6 million positions in the genome, approximately. Then we compared between the patients and the controls, and using statistical tools, mathematical analysis, because we want to make sure that what we are observing is not due to a random event. You can have something happening in the cases at the same time happening in the controls, and it's only randomly that you see it more prevalent among the cases. So what we do with the statistical tools and a lot of other steps that I will not maybe explain here today is that we make sure that these differences are really happening and are not due only to a random event. So for those 6 million positions, we were able to contribute to analyze them, and we found actually an important amount of information that I'm going to explain to you. We found first that many positions in the genome, probably hundreds, are globally controlling the genetic risk of FMD. Those are tiny parts of the risk that is giving FMD. Having just one default, one genetic default is not enough. You need to have a combination with a large number of those genetic defaults to have the risk of having FMD. So this is a very important feature, because even if FMD is a genetic disease, it doesn't mean that it is easy to determine what precisely genetic default is causing the disease. It is a combination of many different genetic defaults. That's what makes it very hard to study, and that's why we need statistical analysis to make sure we are not making a spurious result or describing something that is not real. We also found that many of these genetic variations, around a third approximately, are the same that genetically determine migraine, hypertension, and intracranial aneurysm. Maybe some of you have experienced migraine as being FMD patients. Some patients in France, for example, they come into the clinic because they have migraine, and when we look at their arteries, we found FMD. So it is not completely random that we see this coexistence between genetic determination of FMD and migraine. But this result is very important. It's not only the clinical observation that is giving us this link between FMD and migraine, but it is a genetic one. That means that a person has maybe a combination of genetic default that at the same time is causing migraine and FMD due to the same biological problem that is not functioning correctly in their arteries. Unfortunately, of these hundreds of genetic positions that we see as important for FMD, we are not able to know all of them. So most of them are still to be discovered. For now, we have 5 of them that we are confident are really important for the genetic risk of FMD. And these are specifically important for cells and are involved in building the wall of the arteries. So very briefly, I will explain to you what is the biological function of those genes. So if you look at the artery here, this is a representation of the tube where the blood is flowing. We can zoom in and you can see that there are 3 layers in the arteries. One is the outside part. It's composed of cells called fibroblasts. They are very important to the structure of the artery, but they are like a sort of support, physical support. Then you have the musculature of the artery. These are the media. Cells, part of the media are smooth muscle cells, and smooth muscle cells are biologically very important to cardiovascular health in general, and specifically to the contraction of the arteries. Action is needed to maintain the blood flowing through the different organs. So the renal artery needs to contract to make the blood come into the kidney. And then you have a last layer, the internal layer of the artery, which is composed of only one layer of cells called endothelial cells. These are also very important in general because they are in contact with the blood. But in our case, we found that the genes that we have identified in general, and specifically the 5 that we have described in more confidence from the statistical point of view, are involved in the contraction of smooth muscle cells. So we demonstrate that those cells need or have maybe too much contraction or not enough contraction. This is an open question that we still have to investigate. So what next? What about the other genes? We do need to have an idea about the function of the other genes. And this is part of our investigation currently. Is this contraction of the only important default that causes FMD? We don't know yet. If we know the function of the other genes, we may be able to answer that question. And what about SCAD? Has SCAD the same genetic organization than FMD? I can say that the answer is yes, but we still have to make an effort to understand the genetic basis of SCAD in a more thorough way as we have done for FMD. This is an ongoing work. And can we use this information to treat FMD? Probably, but this needs, again, more investigation. We cannot just go and correct the default of contraction. First, because we don't know yet if it's too much contraction or not enough contraction. We only know that these genes are important for contraction. So to make sure if this is something happening in reaction, for example, to hormones, is it the fact that women have different hormone sense than men that they develop this kind of default in contraction? Again, this is something we need to investigate to make sure that if we develop a new treatment for FMD that is going to correct for this contraction problem, is relevant, safe, and able to do to give more specific treatment than the ones that are described for any kind of cardiovascular disease. With that, I want to thank my team. These are a lot of very young and enthusiastic researchers that I work with them, and also our international collaborators. The genetics of FMD is an effort that comes from interaction between many investigators. The funding bodies, the European Commission is funding our work, as well as many French institutions like the INSERM and also some foundations of research on cardiovascular disease. So thank you for your attention, and please go and discover our work and get in touch if you are interested in our research. Thank you.